Uh, uh, Sir, I have a question. Like,
Yeah, go ahead. While
you
you're
said
asking,
that
I'm thinking.
thousands of like,
Yeah, go ahead.
I am saying like if you said that like 1,000 paper is published, then we say that it is a research, very highly research a topic. Like I just wanted to know like how many papers are published in on a topic, so that then we consider that it is a highly research topic.
Well. That's a difficult question to answer. It is not just the number of papers. Some topics become very fashionable. Cancer research is a subject which has published really thousands and I mean hundreds of thousands of papers. But they are on a very wide range of topics, and because there is no one single cancer and no one single subject in cancer. There are different kinds of cancers and different aspects of cancer which are studied. From the molecular biology aspects to the actual medical aspects, there's a huge range. So, recombinant DNA technology, the process of protein production using recombinants, that is what we are talking about, is the subject that became the most important. You all know this, right? That is when the first breakthrough came on cloning and plasmid biology, and the first protein, that is, the first recombinant insulin was launched by Eli Lilly. You're familiar with this. This is the story which. This is the problem. 1 second, hold on.
Yes, sir, we are familiar with this insulin.
You're familiar with the insulin story, right? And the recombinant insulin story from Eli Lilly, and that is where then people discovered that, my God, come on! We can make literally thousands of recombinant proteins. Today, the number of recombinant proteins which are of therapeutic importance in is not just insulin. There are about 1,000 proteins here. And then, as technology improved and we realized that we could produce these enzymes, these recombinant enzymes, at rates which are far, far cheaper than even normal enzymes. And now E. coli is not only used to produce recombinant enzymes of therapeutic importance, but also of industrial importance, which means that we are now competing with funguses and yeast and the native. There are native strains which produce enzymes. You're familiar with this, right? You've heard of native strains. For example, the most common, which is taught in almost all MSc, is amylase and protease in Bacillus. Amylase or protease or lacase or beta galactosidase or a host of enzymatic. Almost all of them are either fungal or bacterial, which is Bacillus, and amylase, protease, lacase. I can go on. Just a huge long list of industrial enzymes, beta galactosidase. All of these are now being produced by recombinant route as well, because they now compete in terms of productivity. They have far higher productivity. So even though recombinant enzyme production, so that is why, for example, I was talking of plasmid stability. Why does plasmid stability become important? You realize that plasmid stability becomes important primarily because now you are no longer running insulin plants and small plants of 100 liters and 1,000 liters. You are now running 10,000 and 100,000 liter plants. And the moment you're running 10,000 liter or 100,000 liter plants, what is wrong with this software? Can you hear me? Yeah.
Yes, sir.
Hello? Yeah, because I have to sort of switch it off and switch it on because suddenly the writing stops. So the moment you have these many reactors, plants, your N becomes high, the number of generations becomes large. And remember the graph that I drew for you, the probability of plasmid free cell emergence. So when the fermenter runs for these many, and this is N, and this is percentage recombinants, and so obviously you realize that when the number of generations is small, you stop here. And when the number of generations is large, you stop here, and when it's very large, you stop here. So obviously, this is not acceptable. So the moment N goes up, today it's a problem. Something is wrong with this. So when n becomes large, then your number, the probability is wrong. This is going to become serious. I think we are now in a very major glitch. Just give me a minute. No. See.
So I guess it's not a glitch because at times when you're presenting the screen, then like the system tends to hang at times.
This is hanging many times, too many times. Yeah, it's creating too much trouble. I hope it does not. I mean, this is a software glitch in my laptop. I've not been able to get rid of it for a long time. Can you hear me and see the screen now?
Yes, sir.
Yes, sir.
Yeah. Yeah, thanks. At least that much is okay. So right now, so as I was saying that this actually disrupts the class actually. So that as n goes up, your probability of plasmic free cell becomes very, very high. And because of the nature of this graph, you can see that if this graph were to be like this, my job is done. So if this, so the ability to reduce probability of plasmid free cell emergence so that when the fermenter runs to this long, it is still 100% is all that is required. So that is why plasmid stability became important, because of the need to scale up production from 100 liters and 10 liters and in shake flask cultures, which biologists were doing, to engineers who are now moving into 100,000 liter reactors with recombinant enzymes. So there is an industrial demand, there is a real social demand for a certain subject to be studied. Anyway, so any feedback or any questions? We will stop plasmid stability today and move on to the next steps, because we have to talk about the whole production process of recombinant enzymes. Any questions?
Sir, I wanted to ask that in this course we have to study also enzyme science and kinetics. So when will it start? Means.
Well, I want to start it as quickly as possible. And you want me to start it? You want me to wrap this up very quickly? We can wrap it up very quickly. I mean, some amount of I really am under tremendous pressure. You can well imagine I'm juggling too many subjects and I'm trying to do justice to all of them and yet cover many subjects. So I will always be under this continuous stress, and the only way to resolve this stress is by feedback from you guys. Your feedback is, yeah.
So because there are many concepts in enzyme science and kinetics, like mathematical concepts, means which we should like ponder upon.
Yes,
so that.
Sure. But so should you about plasmid stability. Now, I will give you a very simple example on plasmid stability, just because you mentioned it, that mathematical concepts. I bypass those mathematical concepts. Now, think about it. This is a very good question for you people. Just think. Now, what did I write? I wrote the material balance dx plus by dt is equal to mu plus x plus minus d dilution rate x plus, this is wash out, minus p mu plus x plus. Right? This is the plasmid free cell emergence, and dx minus by dt. Do you remember these equations? Both of you? All of you? Mu minus x minus.
Yes, sir.
Minus dx minus, because this is washed out at this rate, plus, and this is p mu plus x plus. So this is the part which is sort of getting converted. So this is minus, this is plus, because this is the part which is forming plasmid free cells. So this is the conversion of x plus to x minus. And I multiplied by p, where I said that p is the probability of plasmid free cell emergence given by p is equal to 2 raised to 1 minus n. Remember this? Now, let us explore this mathematical concept a little bit, not too much, because I don't have time again. Now, first of all, as I said, n is n average. But you cannot do n average when you're calculating probability. You have to have the distribution. So if it is n1, n2, ni, and n is a distribution, so each of these cells would be actually, the probability would be a summation of the probabilities 2 raised to 1 minus n I is equal to 1 to say x, where x is the total number. And that this is not the same as 2 raised to 1 minus n average. You can well imagine that if ni had an average of n average, then this probability will not be equal, because it is in the power index. And so if there is a distribution of copy numbers like this, then these high copy number will contribute very low probability, but low copy number plasmids cells because your copy number has not got corrected. Now, this is very difficult to find out mathematically, and therefore we don't want to investigate it. I mean, experimentally, it's very difficult to find out what is the copy number distribution inside a growing population of cells. So that's why we don't do this, but definitely the probability will be higher than this. This P is definitely, this P star is definitely higher than P, because the more skewed the copy number distribution, the higher it is, because these cells with less copy number contribute hugely to the probability, even though they are small in number. So they give a high probability of plasmid free cell emergence. Now, the second more very simple question that I want to ask you is the following. Now, this is P, right? For all practical purposes, this is P. P is equal to 2 to the 1 minus n. But I have used P here as this P. Are these two P's the same? Or is there a mathematical relationship between the two? Think about it. I am saying that my mu plus, if there was no plasmid free cell emergence, would be mu plus into x plus. So now my mu plus real, because of plasmid free cell emergence, is actually mu plus into 1 minus P. Because P is the fraction of plasmids which are getting converted. But this is actually one cell dividing into daughter cells, and so there are two cells which are supposed to form, and this is plasmid free cell and this is plasmid containing cell. The mother was a plasmid containing cell, and this probability in every division is given by 2 to the 1 minus. So instead of two cells, the mother cell X plus does not give rise to two cells. It gives rise to 2 minus P cells in one division. In one division, it gives rise to 2 minus P cells, and it gives rise to P. Of course, when I said 2 minus P, what I mean is that 1 million gives rise to 2 million cells minus 2 million minus 1, if 1 is the probability of the emergence in 1 per million. So, you know, when I said P is actually 10 to the minus 5 or whatever, or 10 to the minus 6. So it is, instead of this, it gives rise to 1.99 cells, and 0.01 cells are plasmid free. Obviously, it's not 0.01. When I say this, I mean to say that out of 200 cells, 100 cells, when they divide, they will give 199 plasmid containing cells and 1 cell which will be plasmid free cell. So instead of 200, it is 199 and 1, or 199 and 1 if it is 1000, or 1999 and 1, and whatever, you know, that depends on the probability of plasmid free cell. Now this P and this P, are they the same? They are not. Think about it. Now cells divide in a doubling time of TD. Let us say doubling time is TD, and I hope that when cells are dividing in a exponential fashion, you know that mu is equal to ln2 by TD. Are you familiar with this equation?
Yes, sir.
This is easy enough to derive. It's simply by the fact that dx by dt is equal to mu x. So if it is equal to mu x, then you know you can do a integration, bring x down, mu there, and find out between x1 and x2, which is double. So you can derive this expression easily enough. Right? Now mu is equal to ln2 by td, which means in time td, two cells are formed, because you have integrated between x2 and x1, and you have said that the doubling time means x2 is equal to 2x1. Now here in TD, you don't form two cells, you form ln2 minus p cells. So your mu real here, your mu real is actually equal to, what is it equal to? It's equal to ln2
minus P.
P upon TD, and this TD is equal to the mu upon ln2. If mu were the real growth rate, mu plus. Are you getting this point? So this is the relationship between mu plus and mu real, which is ln2 minus P upon ln2. Okay, substitute this back into this, then what do you get? I hope you're following this, right? So what is mu plus real and mu plus relationship? What is the relationship in terms of, tell me, come on. Hello, yeah.
So nu plus real into nu is equal to ln2 minus p into ln2.
Sure, or is it division? Did I make a mistake?
So it's division.
Sure, come on, let us have some versions here. Okay, let us have it.
So mu plus real divided by mu plus is equal to ln2 minus p by ln2.
Absolutely. Mu plus. Okay, I know this is causing confusion. Take all of you as a very small suggestion. This is a bad habit that you
I
guys
guess
develop
for TD you wrote.
All of you have to have a paper and pen, and you have to have your class notes. Just because you sit in my room and in your pajamas and you don't have to bother about it, and you know that this is being recorded, it is a very good habit. And this is a separate thing completely. It's a very good habit to sit down and with a paper and pen and on your table and actually take notes. The process of taking notes is not to refer to them. The process of writing impacts on the brain in a different way. When you write something which you are hearing, then it gets internalized much better. Otherwise, you will listen and you will forget. So this is it was not an old style, old school suggestion that students were asked to come with notebooks and with a pen. The process of writing, I have a very simple example is when you write down somebody's phone number, and because you want to remember, you don't have to refer back to what you have written. But if you listen to the phone number, you tend to forget. Because the process of writing internalizes what you are learning. So I would suggest all of you, as a matter of good learning practice, always have a notebook and a pen. Otherwise, listening is a passive exercise. Writing is an active exercise. And that helps. So that's something else. But you know, mu real is ln2 by. So mu is otherwise ln2 by TD. And mu plus real is ln 2 minus p by TD. Okay, and these two TDs are the same. So you can see what the relationship is between mu real and mu plus. And what I have been using here as p is actually, I have been claiming that this p here is really what we have been claiming is that mu plus real is actually mu into 1 minus p. Isn't it? From this, you can see this. Now, this p, I will call it the p here. I will call it the p. My God, again it is gone. Okay, sorry. I'll just put it off for a second. Can you see the board? And can you hear me?
Yes, sir.
Yes, sir.
Yes.
Yes, sir.
So this P, I will call it as P star, which is what I'm using here, the P star. If I call it P star, and this P equal to 2 raised to the power of 1 minus n, this P and this P are now different. You realize this P is the equation here and this P is the equation here, P star. And this is the equation for P, which is from this expression here. So I would like you to now go back and take this as a very small exercise. Quiz mein aane wala hai. Sorry, that's the only joke that I know, which I feel is not really a joke at all. But so I wanted to say that this P star is a different P from the P that I've been using. And I used it very neatly and I just wrote it and none of you objected. Because, you have to go into the mathematical depths to try to. So this is just this whole thing was just an example to show to you that there are interesting mathematical concepts involved here, which we are neatly sort of sidestepping, because this P star and this P here are quite different. Anyway, let's stop here, because I don't want to, I mean, as you rightly said, we have to go back to enzymes, and and this really wants me to, this fellow is really a problem today. There is an issue here. Okay, can you see the board and can you hear me? Okay,
Yes, sir.
let us quickly move on, because we have very few lectures available. In the next couple of lectures or 2-3 lectures, I will wrap up the whole process of plasmid protein production, which, as I said, is the following. I mean, it is not just the DNA which is contained in the plasmid. The DNA has to be made into RNA, or rather mRNA, and then this has to be converted into a polypeptide inside the cell, and of course this polypeptide has to fold. And then, of course, it has to get exported. Depends on where it is going to go. And each of these steps has been studied in very big detail. And the steps, and then as I will talk about it, these steps are taking place inside a cell. And so the cell itself reacts to this process in a very complex way. And that also has been studied. So right now we were talking about this whole issue that the DNA should be present. If it is absent, then obviously you cannot get a product. And that is the issue of plasmid instability. This DNA is plasmid DNA, because the plasmid contains the DNA of interest. Now, to make it into an mRNA, what do you need? The DNA should have a promoter, right? Now, there has been a huge amount of research on promoters. This is every second now. There has been a huge amount of study on promoters. Can you hear me? Every time I have to ask this, yes, I'm sorry. Yeah. So, as I said, the initial promoters were Lac promoters, straight away borrowed from the beta-galactosidase operon. Okay. And one of the problems with the Lac promoter is it is called a weak promoter. Now, the concept of weak and strong is the following. A strong promoter is what makes a large number of copies of mRNA per unit time, per unit plasmid. So, because promoters were weak earlier, the idea was to use plasmids with very high copy number. To use high copy number plasmids. Why high copy number plasmids? Because even though there are the promoter is weak, even though the promoter is weak, the number of copies of the high plasmid copy number would also mean high copy number of the gene. Gene is also high copy. If the gene is high copy, even if the promoter is weak, sufficient mRNA would be made, and therefore a large amount of polypeptide or protein would be made. And there was the idea that if you can just somehow make a very large number of amount of mRNA, then by definition, this mRNA would get translated. This is transcription. So the assumption behind all these studies was that transcription was the rate controlling step. And it was also essentially true. Is the transcription is rate controlling step. And as chemical engineers and as biochemical engineers, you understand the importance of rate controlling steps in biology. And this has been a very major question. As a matter of fact, are there rate controlling steps A to B to C to D to E in the way we understand chemical engineering, or is biology more complicated? There are no rate controlling steps. And I used to call it, for those of you who might be interested, we had developed in our earlier days of research something called the shifting bottleneck model. I like the term, the shifting bottleneck model, which means that there is a bottleneck to a particular flux of metabolites A to B to C to D to E, and this is part of synthetic biology nowadays, so it's very important anyway. And you say that A to B is rate controlling. Now you do something by which you remove this constraint. Then you will discover that suddenly B to C is rate controlling, or C to D is rate controlling, or D to E is rate controlling. So the bottleneck to the flux of this pathway shifts downstream or upstream depending on which is the bottleneck that you have removed. So it is not that there is one bottleneck, DNA to mRNA, which is the transcription, and the remaining speeds are very, very fast. They are also reasonably slow. Unlike chemical industry, these are rates which are matched. And one or the other rate is slightly slower than the other. And we tend to have an oversimplified opinion in most of our understandings of including enzyme kinetics and multi-step enzyme kinetics, that one of these steps is the rate controlling. When you have rates which are similar to each other, your model becomes far more complex. I hope you are familiar with these A to B to C to D models in your reaction engineering? Have you done this multi-step kinetics? Hello.
Yes, sir.
Hello. You have done multi-step. So, you can always simplify. You can always write differential equations for A to B, B to C, C to D. And these differential equations become correspondingly more complex because now you have to solve for the fact that B is changing because of A and then C is changing because of B. And so the values of B and C can be called as quasi-steady state because they are forming and as well as being consumed, as well as being produced, and so on and so forth. But those are interesting models. But in biology, these models become much more complex. So to come back to the point, a weak promoter was compensated by high copy number of plasmid and hence high copy number of the gene. But the bigger and there was a search for stronger and stronger promoters. So from lac we went on to the promoter. This is crazy. Can you hear? Trip and then
Yes,
a
sir.
combination of tryptophan to TAC, and slowly better and better promoters were discovered. And I will not, as I said, as you people rightly said, let us not get into too much of detail of recombinant DNA technology. And there were a series of promoters which were discovered, which culminated nowadays, as a matter of fact, the most popular being the T7, which I talked about just now, which is a fast promoter, and it is run by the fact that the chromosome contains the T7 RNA polymerase. You have heard of this? I think I mentioned this in the class earlier, in the first class. The T7 RNA polymerase is in the chromosome, integrated into the chromosome under the lac promoter. So it's a two-step induction. The lac is induced, as you all know, by IPTG because it is it mimics a lactose, and IPTG and the T7 RNA polymerase then triggers the T7, and this T7 then binds to this gene of interest and produces mRNA, and a very large amounts are produced. And so it was felt that this is the solution. But remember, we just now talked about plasmid instability, and that is why we need to understand the interrelationships between subjects. What is the interrelationship? The problem is that when you are converting DNA to mRNA, you are also drawing energy away from biomass synthesis into protein production. And therefore, by definition, your mu plus is significantly less than mu minus. Now, how do you take care of this by biological systems? How do you handle this alpha value? And the simplest way to handle this alpha value is to say that I will divide up my fermentation. I will divide my fermentation into two parts. One, the growth phase, and second, the production phase. So what I will do is that I will actually take my fermentation here, and here's the time, and what I will do, and here is, say, let us say biomass X, and I will allow the biomass to grow exponentially. I will allow my biomass. Can you hear me?
Yes, sir.
Hello? Yeah. Can you hear? Okay. And now, if I were to, this is the biomass growing normally, but it is now divided into growth phase and production phase, which means that for a very long time, let us say till this point here, there is no product. The product is zero. And at this point, I will induce the culture, let us say with IPTG, and the product will start forming. Now, this is a very high amount of biomass already, so the rate of product formation would be very high. And the product formation phase, notice, will be run for a very, very short period only. Let's say a bit more. So the production phase or the product formation phase lasts for a small time. Can you tell me why? What is the advantage of such a separation of growth and production phases? Can you tell me this? Hello? Hello.
Sir, we can hear.
Can you hear me? Yeah,
Yes, sir, we
go
can
ahead.
hear you.
You can hear. Yeah. So what is the advantage of a separation of growth and production phases?
So maybe the energy can be used in a better way when the cell is performing one function rather than we're dividing it into two functions
Yeah,
simultaneously.
that's right. That's right. But what practical advantage do I get by the fact that the cell is not stressed for a long time? See,
We get
so
good amount of
this,
biomass.
yeah. So A, we have a good amount of biomass, and so we trigger the production in the later part. So we start producing product, then we really produce product very fast. Now, what is the problem if I were to trigger the production from right away from here and reach the same value? What is the advantage of not producing anything and suddenly producing a lot? Instead of producing it slowly as the cell grows? That is the question that I am asking. And let us go back to our understanding of plasmid stability. What did I say about plasmid stability? I said that there is a concept of emergence, but there is also a concept of overtaking. Alpha is equal to mu plus upon mu minus upon mu plus. And this mu minus is higher than mu plus. Why is mu minus higher than mu plus? Because, just like you said just now, it is because the cell is producing product. So a metabolic flux for product formation, flux is diverted for product formation. If flux is diverted for product formation, then this alpha value is large. But if there is no flux diverted for product formation, the presence of the plasmid inside the cell, the plasmid presence is not a huge metabolic burden. Is not a huge metabolic burden. The real metabolic burden comes from the fact that the plasmid is forced to produce the product. So if I grow the plasma cells in the growth phase, but I don't induce, then the product is not being formed. And therefore, my mu plus and mu minus are very close to each other, which means alpha is close to 1. And therefore, plasmid free cells may emerge, but they will not overtake the culture. In the later part, at this point here, I am suddenly inducing the culture, and therefore my product formation starts. Immediately there is a huge flux diversion towards product, and there is a metabolic load on the cells, which really means that now my mu plus is significantly lower than mu minus. But, and there's a very important but, this time is very short. This is a short time. If this is a short time, even though alpha values are very much larger than 1, and therefore plasmid free cells start emerging, plasmid free cells now start overtaking rather fast, but it doesn't get enough time because the production phase lasts for a very short period of time. If it got the whole time from here to here, it would have definitely overtaken. So the delay in induction, the delay in induction and thereby separating the growth and product formation is again a bioprocess strategy to reduce instability. So always, so now you see how subjects get linked together. So you are now looking for a promoter. You are now looking for a promoter which is not only strong but also inducible. Now, almost all promoters are inducible. So you will say, sir, what is the big issue? I mean, lac is also inducible. Other promoters are also inducible. And now that is why the engineers talk in a different language from biologists. How strongly inducible? Not just strong, but how strongly inducible is the question that we ask. What does it mean? It means the following. It means that you can have a promoter where the non-induced phase and where the non-induced phase is this much, and if I induce it is this much. Or you can have a promoter where the non-induced phase is this much and the clearly this promoter here, this is the point where I induce. So the non-induced, notice that this here non-induced is close to zero expression. This is the expression level. While this promoter here has a basal level expression, even under non-induced conditions. Such promoters which have high basal level expression are called leaky promoters. And leaky promoters would tend to reduce mu plus and hence increase instability. So there is a role between instability and the leakiness of the promoter. How leaky is it that the growth phase allows for protein production to take place, and that lowers mu plus compared to mu minus, allowing mu minus cells to emerge. So we want, instead of leaky promoters, what do we want? We want tightly regulated promoters. So the fold increase in expression between induced, this is uninduced culture, and induced, is a very important measure of leakiness. Because obviously you don't want a very tightly regulated promoter which also does not increase, give expression when it is induced. So the fold increase, for example, with lac is 10-fold, 5 to 10-fold only. Which means that if this is uninduced culture, it's producing so many mRNAs per second, the fold increase is 5 to 10-fold. While on the other hand, even T7, which starts at a high value and goes very high, is 10 to 20-fold. But for example, a PBAD promoter or a Ranose promoter, and Ranose I'll come to later on, all these promoters, they have 1,000-fold increase, which means that they're very low and they go very high. So tightly regulated promoters is the requirement that under uninduced condition there should be no leakiness. Do you realize uninduced condition? There should be no leaky expression. So, and then when it is induced, it should go up 1,000-fold. So how good is the switch? Is the off switch? It is not an electrical analogy, but it's important to understand. In electrical sciences, in electrical engineering, you have a 0-1 and the fold increase is infinite because it is truly zero. In biology, you cannot have 0-1. It is 0.1 and 0.8, and so it is only 8-fold. But if it is 0.01 and 1, then it is a 100-fold increase. So whether it is 0.0, now this becomes very, very important when we are trying to express more proteins and especially toxic proteins. So toxic protein expression has many proteins, and when I say toxic protein, I don't necessarily mean toxic to you. One gentleman came to me when I was a faculty, said, here is a toxic protein, I want to express it, can you please do it for me? I said, will you pay me money? He said, yeah, sure, no problem, 5 lakh rupees for producing the protein. And then I asked him, what is the protein? And he gave me a peptide sequence. And then I told my student, please run this peptide sequence. And it turned out to be the active ingredient of what? Active ingredient of snake poison, of snake venom. Cobra, of nothing else. I said, this is a good idea. Let us produce this peptide. Now, if I have any enemies, we can inject people and say they died of snake bite. But then the students pointed out that if the flask breaks and the media gets into a cut in us, then also there might be something like a jail term for you. So I returned the 5 lakhs immediately and said, I'm not interested in producing this protein in E. coli. Now, the point is that toxic is, of course, not just toxic to humans, but as far as we are talking now, right now, is toxic to the cell, the bacterial cell. And many proteins which are toxic to humans are also toxic to bacterial cells because the mechanisms of toxicity are very similar in terms of binding to DNA or being very positively charged or whatever, and so on and so forth, or blocking the electron transport chain and oxygen uptake. which happens with cobra venom. But so toxic proteins will tend to make mu plus really, really small compared to mu minus. So for toxic proteins, you really don't want to induce it at all. And you want it tightly regulated, and you induce it, the growth rate falls tremendously, but within a very short period, you have made a small amount of protein and your job is done. So the more toxic the protein, the more important it is to have a tightly regulatable promoter. So now, the other point that I will come to say, so PBAD and all, and so how do you get perfectly inducible promoters like the way electrical engineers want, which means that the expression is exactly zero. It is zero when it is uninduced. And people have thought of very, very clever techniques, and I will not get into it too much. I'll just give one example, and that is called the flipase. So you have a promoter like this, and remember your gene is like this. And people put a flipper on this, and this is a enzyme which acts on this and this, and the original position of the promoter is like this. Obviously there is no expression, and there are these two sides, and these two, and when you induce some other protein somewhere else, it goes there and it flips the promoter in the opposite direction. So it flips. And so this is the wrong orientation, it is shut off, and this is the right orientation, it is switched on. So you can actually take the promoter and keep it pointing in the opposite direction of the desired gene, and when you need it, you flip it in the proper direction, and now it gives the expression. So this kind of promoters and clever designs like this, and maybe you should think about these things and study them, are some of those which are truly tightly controlled. I just give you one example. I don't want to give hundreds of very, very clever methods, a tightly controlled expression. For example, one of the things, because people work with T7 RNA polymerase, and T7 promoters are very, very notoriously leaky. These are leaky. So somebody, because everybody wants to keep on working, biologists are very, very conservative people, they keep on working with T7 promoter, it's leaky and therefore not able to express. Somebody has put a T lysozyme, T-lyse, which is called it. It's called P-lyse or T-lyse? P-lyse, I think. P-lyse. The protein lysozyme is coded into it, and this lysozyme then actually has the property of actually degrading T7 RNA polymerase. It's a designed specific lysozyme which degrades and attacks T7 RNA polymerase. So the little bit of T7 RNA polymerase that is produced in the leaky version, which actually causes the leakiness of the T7 promoter, is actually degraded by P-lyse. Yeah.
Sir, I'm not understanding what does it mean by saying promoter is leaky. Like promoter is controlled by operators. Then if operator is not functioning well, then only promoters can, the RNA polymerase can go to the promoter and the transcription can happen. But in case of you are saying that it is a leaky promoter, what does it mean? Like it doesn't, it is not regulated by itself, that when it will start allowed to start transcription and when the transcription will not happen. That is controlled by operators. So what's meaning of leaky promoter?
ये तो आप बड़े फंडामेंटल सवाल पे आ गए, और मुझे पता था कि I thought you people have done some kind of expression transcription control and how induction takes place. Do you remember?
We have learned a bit in molecular biology.
Yeah, you have learned it, right? Now, the promoter is actually a site where the RNA polymerase binds, right?
Yes, sir.
RNA polymerase binds to this site, right? And the degree by which the intensity of binding, the way this promoter sequence is able to attract the RNA polymerase to itself and make more and more molecules bind, and then it takes the and then it sort of captures RNA and forms the mRNA molecule. You remember this? And this RNA molecule cannot be formed if there is an operator region, and there are various ways. There are both feedback and feed forward controls. There are all kinds of feedback controls where even after the RNA polymerase binds to the site. And let us not get into molecular biology. This is a very big problem because I'm teaching enzyme science, but I will sort of, since you asked the question, I will answer this. But later on, I will not answer these questions. These are questions that have to be answered by molecular biologists. We are interested in protein production. We cannot keep going backwards all the time. But I will answer this one. So the operator region then prevents transcription to take place because of the binding of various molecules to the operator region, lactose and so on and so forth. So allolactose, and the essential idea is that IPTG or any inducer either binds with the repressor or essentially it promotes this transcription to take place, right? Okay, and in the absence of the inducer, the repressor is bound very strongly. So the inducer deactivates the repressor. For example, one of the possible examples, it deactivates the repressor, and allows a transcription to take place. Now the point that a leaky expression simply means even if inducer is absent, my repressor is not very efficient, and therefore slow transcription is always taking place. That is all I mean. Okay. For all practical purposes, this is the thing that you need to understand, the details of this molecular biology. So if the repressor is bound very weakly and the promoter is still able to transcribe and make mRNA, then it is a leaky promoter. Because even in the absence of inducer, the expression level is taking place. But when you add the inducer, of course, the repressor is deactivated and the expression takes place faster. So a leaky promoter is like a leaky tap or a defective tap. When you close the tap, you expect that the tap will not leak, but water keeps leaking through. That is a leaky promoter. Okay?
Yes, sir.
Is it all right? Yeah. So the idea of P-lyse is that if the tap is leaking, because there is an RNA polymerase sitting there and expressing it, and a little bit of RNA polymerase is being produced, then produce P-lyse, which will degrade RNA polymerase. So the leakiness is blocked to a certain extent. And P-lyse is used to make the T7 promoter less leaky. So there are various strategies. You can degrade RNA polymerase, you can switch, and these are mechanisms by which promoters are designed to become less leaky, and this allows you to actually then produce the protein and separate the growth and production phases, and thereby growth and production are separated, and that allows you to actually reduce the problem of instability, especially with toxic proteins. So the design of promoters is not just to get strongly inducible promoters, but also tightly controlled expression. So the promoter design is no longer limited to having a very strong level of expression. So they need not just be strong, they should be strong, which means high and tightly regulated. This second part is very, very important in today's world. So a strong promoter is not good enough. It should be tightly regulated or tightly controlled, which means that in the absence of induction, the expression levels should be extremely low. That allows you to produce toxic proteins and reduce the problems of instability. Is that argument clear?
Yes, sir.
Yes,
now,
sir.
yeah, so one last point, and before I end the class, is that not just do you want a tightly regulatable promoter, so you want a strong promoter, and these are things that are not taught in molecular biology. That's why I'm sort of mentioning this is part of engineering. You want a strong promoter, you want tightly regulated promoters, tightly regulated promoters, and you also want titratable promoters. And I will come to the last part in the next class, titratable promoters. So you want a promoter which is not just strong, You want a promoter which is tightly regulated. This is for stability, right? This is for high production. And this is for the reason which I'll talk about now, which is let us first explain what is tightly regulatable. A tightly regulatable promoter is of course, zero expression when the level of, the inducer level here is being increased. And as I increase inducer, my expression, oh God. Can you hear me?
Yes, sir.
So, yeah, so the idea is that an inducible promoter the expression goes like this. This is a titratable promoter where the level of inducer determines the level of expression. So not only do I want a tap which is not leaking, but I want a tap where the degree to which I open the tap changes the water flow rate. You don't want a tap like this. You don't want. So here no expression and here all the expression. The moment I add inducer, all full expression. This is a tightly regulatable promoter, but this is a titratable promoter. A titratable promoter allows you to regulate the level, quantitatively regulate mRNA levels. Why do you want that? Can you tell me? And we'll stop the class here. Why do you think we want a titratable promoter? And Ranos, for example, the Ranos promoter is a regulatable promoter. Why do you want a regulatable promoter? Yes, please.
Want to regulate.
Any answers? Because remember, I talked about DNA to RNA to protein and all that. I just now talked about that. And I said that this is the rate controlling step, polypeptide, and this is the rate controlling step. But it may not be the rate controlling step. So there's no point making an expression very high if this step is slow. Just making useless amounts of large quantities of mRNA is not the purpose. So a tightly regulatable promoter will ensure that this no longer remains the rate controlling step. So enough mRNA is made so that this is not the rate controlling step, which really is determined by downstream steps. And we will talk about matching the upstream and downstream steps so that this whole pathway runs very smoothly and you don't have just unnecessary accumulation of mRNA. So we'll talk about this in the next class. Within a couple of weeks, as I said, we will wrap up this whole process of protein production and go on to enzyme kinetics. Yeah, but we have enough time for that. Don't worry. So let us not always talk of other subjects. So any questions? No. When we come to enzyme kinetics, we'll talk about that.
Sir, what is the book for this recombinant?
मैंने बहुत ढूंढा और मुझे मिल नहीं रहा है. I'm very, very sorry. I will definitely try to locate it. Books on recombinant protein production are usually either too advanced or too much of molecular biology to be of interest. So I have located a few books. None of them are to my liking. I am trying to look for more. I think I'll start looking in the net and start trying to download things because all the standard textbooks are not to my liking at all. I mean, they do not have the perspective that I want to push into you, that of a biochemical engineer who uses molecular biology as a tool. It's all full of molecular biology text and long detailed descriptions of plasmids and how they are constructed. Precisely what I don't want. So, sorry, give me some more time.
Sir, in that case, you can provide some research papers which we can understand.
भाई वो तो और मुश्किल हो जाएगा आप लोगों के लिए. मैं दे सकता हूँ. I have no problems. I can give you review papers. Okay, let me see. Let me pass on a few review papers. You people can go through them.
Or sir, you can give us a homework type of question paper that the mathematical questions like 2, 1 minus n and probability finding questions so that we can solve all the questions, and if we have any doubt then we can ask you in the class.
Okay, I'll do that. Okay.
Yes, sir. Problems will be better. Like in that case, we know in which direction we have to learn.
Okay, fine. Thank you. So,
So one
thank
more
you very
thing,
much. Stop recording.
so-
Yeah.
So one more thing. So regarding the lab, so since our batch is